Sathyabama Institute of Science and Technology.
Dear all, this is Dr. S. M. Indhumathi from the Department of Biotechnology, School of Bio and Chemical Engineering, Sathyabama Institute of Science and Technology. Today, I am here for a presentation on the topic Hematopoiesis and Cells of the Immune System. So, hematopoiesis. We define hematopoiesis as the formation of blood cellular components from the hematopoietic stem cells that originate from the bone marrow. So, in a human adult, roughly around 10 billion to 100 billion blood cells are formed every day. Hematopoietic stem cells basically they reside inside the medulla of the bone marrow. So bone marrow is nothing but a spongy cancellous tissue that is present inside the medulla of the bones, and the bone marrow is dark red in color, which contains the hematopoietic stem cells. These hematopoietic stem cells are basically self-renewable, which means in a pool of hematopoietic stem cells, only a set of cells actually differentiate into blood cellular components, while the other set actually remain inside the bone marrow for future differentiation of blood cells. And this type of division is called asymmetric division. So, this hematopoietic stem cells basically divide themselves into two lineages, the common myeloid progenitor and the common lymphoid progenitor. So, this is the process of formation of blood cells. As you can all see, it starts from the multipotential hematopoietic stem cells, which will actually differentiate themselves into two larger progenitor cells, the common myeloid progenitor and common lymphoid progenitor. The common myeloid progenitor gives rise to varieties of cells like megakaryoctytes, erythrocytes, mast cell, myeloblasts, and you have the common lymphoid progenitor that gives rise to natural killer cells and small lymphocytes. So coming to the cells of the immune system, as we all saw, the hematopoietic stem cells gives rise to the common myeloid progenitor and the lymphoid progenitor. We'll first talk about this common myeloid progenitor. It starts with the megakaryoctytes. So megakaryoctytes, as the name indicates, they are the largest of all the blood cells, around 10 to 15 times larger than other blood cells. The size ranges around 50 to 100 micrometer. The only function of megakaryoctytes is the production of thrombocytes or blood platelets. They take the help of a protein called thrombopoietin. Coming to RBCs. So RBCs are red blood corpuscles, as we all know, and the formation of RBCs is known as erythropoiesis. The actual function of RBCs is to take up oxygen from the lungs in case of humans and gills in case of fishes and supply it all over the body. This hemoglobin, which is present inside the RBCs, are basically able to bind with oxygen and tend to supply it all over the body. And this hemoglobin gives the red pigment to the RBC cells. We have around 10,000 RBC cells that are produced every second, every day. And inside a single RBC cell, we have around 270 million hemoglobin molecules. RBC cells are circular biconcave discs without prominent nucleus. Coming to the mast cells. So the mast cells are basically a type of granulocyte cells which contains granules rich in heparin, histamines and serotonin. This triggering of mast cells are usually seen during anaphylactic and allergic reactions. Whenever a foreign particle enters inside our body, the mast cells are triggered and the granules are released from them, leading to the degradation or destruction of the foreign particles. So these granules that are rich in histamines and heparins will reach the site of infection and it will start the process of inflammation. Inflammation is basically a part of body's immune response where the body separates the site of infection from the rest of the body. As we have all seen, an inflammation starts with edema, redness and swelling. It will have a fibrin barrier or a fibrin wall that separates the infected site from the rest of the body. When there is an inflammation, the blood vessels dilate, leading to a lot of blood flow to that particular area, and we'll see exudates coming out of an inflammation. So the exudates are usually rich in either the microorganisms or their products. So mast cells are highly involved in producing this type of inflammatory immune defense mechanisms. Coming to myeloblasts. Myeloblasts are a set of cells which give rise to the granulocyte series, namely the basophils, eosinophils and neutrophils. They are actually very smaller cells which contains a oval nucleus. So we'll first look into basophils. Basophils and mast cells are more or less similar, except for their nucleus. The nucleus of basophils are usually segmented, while the nucleus of mast cells are usually lobated. They have a spherical or an oval nucleus usually, and basophils are usually found in circulation, while mast cells are usually found in tissues. But the exact function of mast cells and basophils are similar. Basophils also have the same granules that are rich in histamines and heparins. They also initiate inflammatory immune defenses. And these basophils are the least amount of granulocytes present in the body, and they constitute around 0.5 to 1% only, and they are the largest of all the granulocytes. Then the neutrophils. Neutrophils constitute around 40 to 70% of all the WBC cells usually. They are also containing granules, but they don't rely on the granules for their immune defense mechanism. These neutrophils are actively phagocytic. In fact, they are the first responders for any kind of trauma or environmental exposures. They are actually called as the hallmark of acute inflammation. When we have an immediate infection, when we have a sudden infection or trauma, neutrophils are the first cells that will actually reach the site of infection through a process called chemotaxis. Chemotaxis is nothing but the movement of phagocytic cells towards the site of infection by means of chemical signaling proteins. We have certain chemical signaling proteins called cytokines or lymphokines. We have so many types in that. We have interferons, leukotrienes, and so on. These chemical signaling proteins usually send messages to the phagocytic cells to attract them to the site of infection, thereby starting the process of phagocytosis. So neutrophils are the first cells that will actively perform this phagocytosis function. Coming to eosinophils, these make up around 2 to 3% of the whole of WBC cells, and eosinophils are usually seen during allergies and anaphylactic reactions. So when we go for a differential leukocyte count, if we tend to see more amount of eosinophils in our body, it indicates that we have an allergic reaction going on inside our body. And eosinophils are more related to this IgE antibody, which is also called as the allergic or reagenic antibodies. Coming to monocytes, this is the largest type of leukocyte ever known, and these monocytes are usually amoeboid in structure. And these monocytes give rise to the two major important cells of our immune system, the macrophages and the dendritic cells. So macrophages are basically of two types, tissue macrophages and blood macrophages. Blood macrophages are smaller in size, around 8 to 12 μm in diameter, while tissue macrophages are a bit bigger, 15 to 20 μm in diameter. Blood macrophages are short-lived, around 3 days to a week maximum, and they are mostly effector cells, while the tissue macrophages live for longer, that's months and years, and they serve for the immunological memory. Macrophages are the most important cells that will actually perform the process of phagocytosis. So phagocytosis. What is phagocytosis? So this is a process of cell engulfing. When there is a foreign antigen that is entering inside you, the cells of the phagocytes, that is the phagocytic cells, will actually put their pseudopodia around them, and they start to engulf the cell inside its vacuole, which forms a complex called phagosome. And this phagosome actually makes another complex with lysosome, making the phagolysosome. This phagolysosome actually has certain enzymes which are actually going to degrade the bacteria or the foreign particle that's been taken inside. So the hydrolytic enzymes, especially lysozyme that is present inside the phagocytic cell, will actually degrade or destroy the foreign particle that's been taken inside, and the degraded bacterial cells or foreign particles will be released outside as debris. At times, macrophages also perform antigen presenting also. Then coming to dendritic cells, they are mostly pleomorphic cells which have needle-shaped projections on their cell surface. They usually have MHC class 2 antigens on their cell surface, and these dendritic cells perform the important function of antigen presentation to the T cells. T cells are cellular cytotoxic cells which are not able to immediately initiate any cellular cytotoxicity by itself. Rather, it takes the help of dendritic cells, where the dendritic cells actually takes up the foreign antigens on its cell surface, goes to the T cells, present it to them, so that the T cells will easily identify them and start to perform the cytotoxic activity. So dendritic cells could be simply defined as APCs or antigen presenting cells. Then we are coming to common lymphoid progenitor. The common lymphoid progenitor includes the NK cells and the small lymphocytes. NK cells are usually termed as natural killer cells, which come under the category of large granular lymphocytes. Usually large granular lymphocytes comprises of three sets of cells. One is the NK cell, another one is the ADCC, antibody dependent cellular cytotoxic cells, and the LAK cells, which mean lymphokine activated killer cells. All these three are NK cells only, but they have minor differences in their activity. NK cells are basically, they do not require any antigen or antibody for its activity and cytotoxicity. They are highly active whenever they encounter any viral antigen and during malignancies and tumors. The NK cells which are antibody dependent, which means those set of NK cells, they take up the help of IgG antibody and they show their cytotoxicity, and they are antibody dependent cellular cytotoxic cells. And then we have LAK, lymphokine activated killer cells. They are also set of NK cells, but they take up the help of the cytokine called interferon 2. Only when it is augmented or complemented with the interferon 2, these LAK cells are able to show its cytotoxicities. Coming to the small lymphocytes. Okay, first we'll know what lymphocytes are. Lymphocytes are usually small spherical cells that are present in our peripheral blood, lymph and other fluids actually. So lymphocytes are basically classified into three types based on their size: small, medium and large. Among these three, the small lymphocytes form the maximum component, and the small lymphocytes divide themselves into the T cells and the B cells. While you have other two sets of lymphocytes which are short-lived and long-lived. The short-lived lymphocytes usually live for days, and they are the immune effector cells, which means they are the first responders to any foreign antigen. They will actually initiate the cell-mediated immunity or the humoral immunity, while the long-lived lymphocytes are immunological memory cells which retain the memory of the previous antigen, and it will not show any latent phase during the exposure of the same antigen for the second time, which this is called second exposure. Usually when a foreign antigen enters inside us, our immune system goes into a phase of latency, which means our immune system takes some time to produce antibodies so that the memory cells keeps this in its memory. When the same antigen is encountered for the second time by the same body, by the same host, our immune system will not actually take much time to respond to it. So for this purpose, we have the long-lived lymphocytes. So as I said earlier, the small lymphocytes divide themselves into the T cells and B cells. So B cells are basically Bursa dependent or bone marrow dependent cells, we say. They have B cell receptors on their cell surface. They get divided themselves into the plasma cells and memory cells. B cells are actually responsible for humoral immunity by the production of antibodies. So the plasma cells are the immune effector cells which produce antibodies in immediate response to a foreign antigen, while the memory cells are those kind of long-lived lymphocytes which keeps the foreign antigen in its memory, and when the same antigen is encountered by the host for the second time, the memory cells will actually trigger the body to immediately produce the antibodies, and it's not going to take much time. Coming to the T cells. T cells are basically thymus dependent cells. They are actually produced in the bone marrows, but they reach the thymus to mature there. They have T cell receptors on their cell surface. These T cells are divided into three major types. One is the CD4, CD8 and regulatory T cells. CD4 cells are basically helper cells which will actually augment, or it will complement the effect of CD8 cells, which are cytotoxic cells, and they will also help the B cells to produce more antibody. That's why you term it as CD4 helper cells. While we have the CD8 cells which are usually cytotoxic, or they are the killer cells which do not need the help, or it will directly get involved in doing this cellular cytotoxic activities. So the regulatory T cells. Regulatory T cells are nothing but those which are able to differentiate the self and non-self antigens usually. They are also called as the suppressor T cells, because we all would have known about this condition called autoimmune disorders, where our body cells will think the antigens present on our cell surface as foreign. So that's a worst condition which a person could experience, and you have so many autoimmune disorders like myositis, myasthenia gravis, and so on. So for this purpose, the regulatory T cells serves us, which means these T cells actually suppress our bodily cells from recognizing our own antigens as foreign antigens. So the proper regulation of self and non-self antigens will be done by this regulatory T cells. So that's all about the hematopoiesis and the cells of the immune system. I've added the references here, and I've mostly referred this textbook of microbiology by Jairam Panikkar and Anantha Narayanan, and it's also from molecular cell biology of Lodish and Harvey. Thank you, students, for patiently watching the presentation.